Hi, so I'm going to tell you about a new typing scheme for pneumo we've been developing over the past year called GPSCs. So I'll briefly go over why we developed it, how it works, why you might want to use it, and how to use it. So just briefly to say why we might want a typing scheme. So I think the most important reason is that we're going to want to split the pneumococcal population into parts, and we want to make sure that we refer to those parts by the same name, so we all know we're talking about the same thing. Once we've got that, we're going to want to be able to do rough phenotype associations within those clusters. So if we know a strain or a cluster, we might know that it's more likely to be drug resistant, more likely to cause disease. So just by getting the type, we'll immediately get some information about its likely phenotype. We can rule out transmission events between different types, and also just a very convenient way to partition the population and do sub-analyses. Obviously, serotyping has been very popular and in pneumo particularly, but I just want to point out that it's only a small part of the genome which isn't always vertically inherited. So typically we want something that looks at both genetic background as well as serotype. So what would we like of our typing scheme? Well, as I said, we need our names to be consistent between clusters, but we would like our clusters to be useful. We want to be able to create subclusters within our clusters. We want it to leverage all of the genomic diversity in the pangenome to get the best resolution possible. We want it to be fast, so we can use all the available genomes, especially if there's so many now. With so many genomes, many of them will have sequencing errors or gene calling errors, so we need to be robust to those. And we need to have good support in software, so it's easy for lots of people to run and use. So thinking about this, when sort of faced with tens of thousands of genomes, we were deciding what's the best method to use. Things like the model-based, like BAPs, fast BAPs, are sort of out because they're not a proper typing scheme, and they do something slightly different. Gene-by-gene methods, though, are typing schemes, but we were slightly cautious because of the amount of curation acquired, and also the sensitivity to errors in genes and gene calling. Particularly sort of going chronologically forward, 7-gene MLST has too many clusters because of an unfortunate choice of one of the genes in the scheme. Its resolution is also obviously limited, so there's no subclustering. Clonal complex is out because the names aren't consistent. Whole genome or core genome MLST is a nice advance, but we found that it was likely to be too slow, still not be particularly robust to errors, and also that the clusters you got out of it weren't actually particularly useful for the analysis we wanted to do. So that led us to develop a bit of software called PopPunk. So the way this works, very briefly, is it uses k-mers of different lengths, compares them between genomes, and using those can very quickly calculate core and accessory distances without needing to do alignment. So for many species, if you plot those out, you get something that looks like this in the core and accessory space, and you can use machine learning methods to cluster those. And what we see here is that the light blue cluster nearest the origin are those that should be within the same cluster. So I connect those together in a network and my clusters pop out of that. So that's great. Pneumo, unfortunately, is slightly more complicated. So on a smaller data set, it looks a bit like this. So we identify some components that are different, but we don't have a clear sort of set of points near the origin which are clearly within a strain or a cluster, because we've got recombination making this more continuous. So where should we draw this boundary? Before, with this is Listeria monocytogenes, it's quite clear where the boundary should go, but here on the right in pneumo, there are a number of different places that seem like there would be reasonable places to draw the line between within or between clusters. Fortunately, it's turned out some recent analysis in another group that actually you can put the boundary anywhere and it will represent evolutionary history. So that sort of means we are free to choose a position which gives us clusters which are useful. And the way we do that is by something we call model refinement. So we move that boundary around and look at different properties of the network to try and produce clusters which are useful. So originally we did this, which was making clusters as sensitive as possible, so splitting them up into lots of pieces while still keeping them transitive, which stops them being sort of stringy and linking different clusters together, which you can see on the left. So this worked quite well. As the data's got larger, we found problems like this, where you could have one sample collecting lots of clusters together. So we also now minimize betweenness, and that solves that issue. So this is what it looks like on the GPS dataset, so with sort of 10,000 genomes. So you've got a blur here. It'd be unclear where to draw this line, but through optimization, we find a good boundary here. So it's also a useful time to draw a distinction between pop-punk, which is the method, general method for creating typing schemes in microbes, and global pneumococcal sequencing clusters, which is the pop-punk scheme for Streptococcus pneumoniae, and using sequences from the GPS project. So GPSCs are the scheme you want to use in pneumo. They're numbered in increasing order. As their numbers go up, they become less common. Currently, we include about 42,000 genomes from 77 countries in about 1,000 clusters, the largest of which has 2,500 genomes in. We iterate versions of this forwards. Versions add new genomes and potentially new GPSCs. If you're familiar with clonal complex, the resolution is similar, but we don't have the sort of disadvantage of inconsistent definitions. It's faster and all the stuff I sort of said at the start. So why would you want to know GPSC? Well, there's lots of resources available is one answer. So Rebecca Gladstone made fandangos and microreacts. So you can look at recombination, you can look at phenotype like drug resistance, you can look at subclusters, you can look at global sampling for all of the different GPSCs. So it gives context to your smaller set of genomes. You can look and see sort of what the global picture is in these sequence clusters, which is really useful. So if you want to use these yourself, if you've got a lot, you just want to use the command line. You don't have to do any model fitting. It's just a single command. We've got that on the Pneumogen website. You can do that sort of for thousands at once pretty quickly. If you've just got a handful, you can look in Pathogen Watch. So you can drag and drop your genomes in. You'll get GPSCs assigned, but you can also browse what's in there by GPSC and find more information about each GPSC one by one. Coming soon, we'll also get this done in the browser. We've got Marie is working on a bit of software called Bebop, which will automate this process, all run in the browser, pretty much, very little data transferred, not sort of taking your genomes away from you. Does the analysis on your own machine, looking both at strains, so the GPSC, subclusters, serotype, AMR, and so on. And also including sort of the entire context of the GPS project. So in summary, we can come up with lots of different cluster schemes with various different advantages. We want to pick clusters which are useful, clusters which reflect biology, have practical advantages. We think that GPSCs do this better than gene-by-gene approaches, particularly MLST. It's fast, many interfaces, we keep the database up to date, name's consistent. If you want to use other cluster schemes, you can still get MLST out of it, so it's not sort of either or, you can use both. And it'll scale to the future as we get more and more genomes available. A few different methods to assign them. I'll put some links here. I'll put this, if this is of interest to you, this will be on my website later, so you can get these links, look at the papers. I'll just end by thanking Nick, who I worked on pop-punk with, Marie, who's developing Bebop, and Steph, who does most of the hard work in maintaining the GPSCs.